Greetings from the Amazentis Global HQ here at the Swiss Federal Technology Institute Innovation Campus at EPFL. I'm Federico Luna, I'm the Chief Marketing Officer of Amazentis, and I'm delighted to welcome you to the first webinar we're doing for early adopters on the science behind Mitopure and timeline. Today you'll be hearing from our Chief Medical Officer, Dr. Anurag Singh, and Principal Scientist, Dr. Penelope Andrew, about the science and the clinical evidence that we've accomplished over the last decade. We're, as a company founded by scientists and doctors, we employ the highest rigor of science and clinical research, but we also understand that the ultimate test is getting this into the real world and hearing from you on how this is translating in terms of your everyday health. And so today you'll be hearing about mitochondria and how that impacts how we age. You'll be hearing more about the science, the deep science behind Urolithin A, and the clinical evidence around Mitopure. Once the presentation's over, we'll have a Q&A session. You'll be able to ask questions throughout the session, and then we'll be answering those questions at the end after the presentation has been concluded. In terms of how to ask those questions, you'll notice that there's a Q&A function. You simply go in there, you type in your question. You can also upvote questions that you're most interested in. The other thing you can do is if you'd like to leave a testimonial in terms of your experience with Mitopure, you simply go into the chat function and you can type that in. And with that, I will pass over to Dr. Andrew.
Thank you very much, Federico. So today I will start by giving you a brief introduction about a very important organelle that we are going to talk a lot about today, which is the mitochondria. So as many of you may already know, mitochondria are really the power generators of the cell. This is where we produce ATP, which is the energy currency of the cell. So some organs are richer than others in mitochondria. What you can see here on the right is the organs that contain the highest number of mitochondria per cell. And not surprisingly, these organs actually need a lot of ATP to function. So for example, when I'm doing that simple movement, a contraction of the biceps, I am burning millions and millions of molecules of ATP. And it is the function of the mitochondria to replenish that level of ATP and to make sure that there is a constant supply to allow for movement. So as you can understand, mitochondrial function is really important to maintain proper cellular health and ultimately organ function. But unfortunately, what happens over time is that this mitochondrial function was found to decrease. If we look at a picture of mitochondria taken by electron microscopy in a healthy and young subject, which is what you can see here on the left, you can see clearly the outer membrane of the mitochondria. There is a clear structure of what we call the cristae, so the little spaghetti that you can see inside the bubbles. And as we age, you can see that the picture on the right becomes a bit more blurry, and these damaged mitochondria are actually associated with a lower efficiency to produce ATP. And what has been shown over the last decades and decades of research is that damaged mitochondria is associated with a number of conditions, such as loss of muscle mass and strength, obesity and metabolic syndrome, but also cognitive decline. And so coming back to this, if we focus now on muscle, and if we were to measure muscle function over a lifetime, what we would see is that muscle function peaks when we reach around the age of 30 to 40, and then unfortunately already starts to decline until the end of our life. And so what we are willing to bring at Amazentis is an early intervention strategy in order to prevent that decrease as much as possible. And so how do we achieve this? Well, we think that we can achieve this by focusing on muscle quality. For the last years, the main focus of the strategies to improve muscle function has been on the muscle mass. And what we believe and what we are bringing with Mitopure is actually a new paradigm and a focus on muscle quality. What you will see throughout this presentation is that thanks to Mitopure, we are able to target mitochondria, and that this turns into an improvement in muscle quality, in energy production, and ultimately in an improvement in functional and physical endurance endpoints. So coming back to the basics, where is urolithin A coming from? So one thing that is important to remember is that urolithin A is not found in food products. What we find in certain food products are the precursors, which are big molecules called elegans. They are present, for example, in pomegranate, in berries, in nuts. And what happens when we eat these fruits or nuts is that elegans are transformed by the gut microbiota into metabolites, including urolithins. However, as I mentioned, not everybody has the right microbiome to produce it, and when you're able to produce it, it's at variable quantities. So as of today, we estimate that only 30 to 40% are able to produce urolithin A. And so what our approach has been at Amazentis is that instead of giving the precursor and hoping for a conversion into urolithin A, let's administer directly urolithin A in order to unlock its potential. So what you will see in the next slides is how urolithin A is able to induce mitophagy, and I will explain a bit more about that pathway. So here what you have on that scheme, what we could call it the life cycle of a mitochondria. So mitochondria are not static. They move, they change their shape, they evolve. What happens over time is that mitochondria accumulate damage by free radicals. This is because to produce ATP, they need to consume oxygen, and that eventually, unfortunately, that oxygen molecule can be converted into free radicals. So that will lead to the accumulation of damage, which is represented in blue here. But this process is normal, it is natural. And the cells have mechanism to repair these damaged mitochondria. First, they isolate the damaged mitochondria by a process called fission, and then digest it by the process called mitophagy. And what we have found is that Mitopure is able to trigger specifically mitophagy. Now, when mitophagy is triggered, this releases the building blocks of the mitochondria, which are represented here as little blue bubbles. And these building blocks can then be reused in order to reassemble new mitochondria, which is a process called mitochondrial biogenesis. And so if we come back to these organs that contain more or less mitochondria, that is an important concept, because cells want to maintain always the same number of mitochondria. So that means that if we trigger mitophagy, the cell will then respond by activating mitochondrial biogenesis in order to maintain a stable number of mitochondria per cell. And so this is exactly what we have seen with Mitopure when it was given to worms. There was first a trigger of mitophagy and then an activation of the biogenesis process. So this is important to keep in mind when you will see later in the presentation our clinical results in the muscle. So there has been a lot of excitement in the scientific community for urolithin A in the last years. I would say that before 2016, which is the year where we published our paper, most of the publications on urolithin A are looking at the precursors of urolithin A or giving urolithin A just to cells in vitro cultures. And then in 2016, we were really the first to show that it was possible to administer urolithin A directly to preclinical models, and that this would have these great benefits on muscle function. And then this has raised the attention of the scientific community on that molecule. And this is, I think, why we see a growing number of papers on urolithin A since then. So if you want and are interested, I really invite you to go on pubmed.gov and to check for all the papers on urolithin A. So how did this all started? Well, it started with a very small animal that is called C. elegans. So this little worm is a very well-established model of aging. It is very convenient to use because it lives for only 30 days, which allows us to know very rapidly if an intervention has an impact on lifespan or not. And so what we have seen with urolithin A in the worms is that worms live about 40% longer. So they have really an extended lifespan. But most importantly, they have also an extended lifespan. They are able to maintain a better mobility. So what you can see on this video here are worms that are 16 days old, which correspond to about 60 to 70 years old in humans. And what we can clearly see from the video is that the worms treated with Urolithin A on the right are really more active. And you may believe it or not, but the video on the left is actually on. These worms are still alive, but they are not able to move a lot. So in worms, we clearly saw with Urolithin A that we have an extension of lifespan, but also of health span. And what was remarkable is that these effects actually translated into bigger animals, into rodents. So on the left, we tested first aged mice that were treated for about 8 months with Urolithin A, admixed into their food, and we measured the grip strength and the running distance. And in the two tests, we saw a clear and significant increase in both muscle strength and endurance after treatment with Urolithin A. And what was even better is that when we went to young rats, so you can see here on the graph on the right that the rats started the treatment actually before 7 weeks of age, which is very young. These rats had access to a running wheel twice per week. This is every dot on the line. So this would correspond to, let's say, bi-weekly training or exercise. And when these rats receive Urolithin A in combination with exercise, we can see really a clear improvement into the running distance. So all in all, what we have shown in that publication is that Urolithin A induces mitophagy in the muscle cells, that this results in an improvement in muscle function, and ultimately this translates into an improvement into the muscle function of these different animals. And so based on this data, we were very excited to see if it would translate into humans, and this motivated the initiation of our phase 1 and phase 2 studies. And so with that, I will leave now the floor to our Chief Medical Officer, who will tell you more about our latest clinical results.
Thanks, Penelope. So what we have done over the last 5 years is really engage in multiple randomized double-blind placebo-controlled trials in a range of healthy populations, starting from elderly to middle-aged and even young populations. So building off the preclinical evidence, what we really saw, and the first thing we wanted to show in humans was really the safety, bioavailability, and the first proof that Urolithin A and Mitopure were working at the mitochondrial health level in the skeletal muscle. So we engaged in the first in-human clinical study, which was published earlier last year. And the way these studies are done is that participants are enrolled based on a bunch of inclusion-exclusion criteria, one which is being they're healthy and they agree to not consume other mitochondrial focused supplements during the course of the trial. They come to a hospital-like setting and they're monitored over a course of 36 hours by 2 trained and qualified MDs who are really looking at a battery of safety tests, starting from ECG recordings, blood biochemistry, and really looking at the adverse events over the course. And so we started with the first cohort of 12 elderly that were given 250 milligram of Mitopure or Urolithin A. Now we really wait to show the safety in this cohort before we escalate to the next higher dose. The length of administration was 4 weeks or 28 days, and we took muscle biopsies at the start and at the end of the 4-week intervention, and as well as blood draws. Once we showed the safety of the 250 milligram, we escalated to 500 milligram of Mitopure, and the last dosing we did in this study was the 1 gram Mitopure. So what are the key takeaways from this trial? As I mentioned, that was published in Nature Metabolism last year. If you want to really read up more, I invite you to look up that publication on Urolithin A and Mitopure's effects on mitochondrial health and safety. In short and summary, what we saw was that Mitopure is very safe. It's a natural molecule. It has been present evolutionary, derived from our diet since our evolution, and it was very well tolerated at all doses. We saw that the blood levels were increased as we increased the dosing over time, and the steady state levels were maintained even in those who had very little or low levels of Urolithin A coming into the trial. Remarkably, we saw in as short as 4 weeks, we were able to stimulate the mitochondrial health and mitochondrial biogenesis in the skeletal muscle of these healthy elderly subjects. And so in the next slide, what you're really seeing is a panel, is a graphical representation of muscle biopsy mitochondrial gene signature before and after the 4-week intervention. So the first panel, let me walk you through it. You see 3 columns. One is a column of placebo subjects. So each column is an individual subject. Each row is an individual mitochondrial gene. And so what we are really looking in terms of change is the upregulation of mitochondrial genes, and which reflect that mitochondrial health is improving in the skeletal muscle of these subjects. So blue, if you focus on the color scheme, blue means that there is no change before and after 4 weeks of intervention, and red means there's a dramatic increase in upregulation of mitochondrial genes and mitochondrial health improvements. So what you're seeing in the placebo, as you see between all the subjects, is mostly blue, which means there hasn't been much of an impact on mitochondrial health in the skeletal muscle. And as you go from left to right, if you focus in the middle with the 500 milligram of Mitopure supplementation for 4 weeks, you start to see that the mitochondrial health and cellular health in the skeletal muscle of these subjects is improving. And then if you focus on the right, you really see a dose-dependent increase, a dramatic increase in the mitochondrial health. We have also in some other trials looked at age-matched elderly who exercise versus those who are very sedentary, and we see similar mitochondrial upregulation in those who exercise over a long period of their lifetime. Building up these 4-week trials, what we did next, and these are recent unpublished data, is we engaged in a much longer 4-month intervention. So we gave Mitopure, we did a randomized double-blind placebo-controlled trial where we gave either Mitopure in different doses or placebo for 4 months. We selected subjects that were 40 to 65 years of age. They were healthy, but they were sedentary and a bit overweight. And so comparing these 3 subjects, we did a battery of tests that are available to clinicians to measure muscle and physical performance. And what we see is really remarkable. So on your left, again, the way this test is done is that these people are coming into a hospital-like setting, and they are immobilized on a chair, and then you're really focusing on the muscle strength and the muscle contraction of a select group of muscles. So they're doing a set of muscle extension or an extension of the leg and flexion of the leg, and what we are seeing is the peak force of the peak muscle strength during this muscle testing that is being taken place. We focus on a group of muscles in the leg that is key to mobility and human movement called the hamstrings. And so if you focus on the middle panel, which is really the data from our randomized double-blind control study, is that you see the axis is basically the change in muscle strength over 4 months of time, the intervention. The placebo group doesn't change. In fact, it even declined in over 4 months. 4 months in human health span is a pretty long time. And when you compare the placebo to the Mitopure groups, the 500 milligram or the 1 gram UA, you see this dramatic increase, which is statistically significant in improving muscle strength in these non-trained sedentary population. This is really remarkable. And when we took muscle biopsies from the same group of subjects and compared the mitochondrial health or the muscle health of in this select group of skeletal muscles, what you see again if you focus on the right panel, going from red color to blue red means upregulation. You're really seeing the upregulation and increase in mitochondrial and muscle health promoting genes, which means that we are having very targeted effects on muscle health that are translating to improvements in muscle strength in this population. So going to the next clinical trial, which was really the question that everyone was asked, what are the levels that you can get from diet, or if you drink, how many cups of pomegranate juice do you need to drink to get enough Urolithin A in diet? Well, the first thing we did was we investigated a whole panel of supplements and food products on the market today for levels of the precursors. These precursors, as Penelope mentioned, are called as phenoclagens or elegans, and we wanted to ask what is the level of exposure people are in the healthy population are getting once they consume these products. So you can see in blue is the level of the precursors, which are called phenoclagens, In red is the Urolithin A or the Mitopure. What you can appreciate is that first is the huge variability in the levels of the precursors. There are supplements which have a lot of the precursors, and then there are others that have basically almost nothing in terms of, and when you look at the red panel, Urolithin A or Mitopure is really present in the timeline product, and none of the other products had any level of detectable levels of Urolithin A. So this is a question that has come to us a number of times when we speak in scientific and clinical meeting is how much glasses or how much cups of pomegranate juice do I need to drink to get enough of Urolithin A through my diet? And this is a question we know there are about 30 to 40% sort of people who have the right gut microbiome, but we haven't really answered it. So this is a recent study that we just wrapped up. We took, this is done in the US in a metropolitan city area, and we took and did the study in a lot of subjects. We took 100 healthy subjects, 20 to 80 years of age, and we randomized them into two groups. Half of them took the pomegranate juice, which is 100% pomegranate juice, as I showed you, that has a high level of precursors. And we took 3 blood draws before they took the pomegranate juice, 6 hours after, and 24 hours after they had taken the juice. Another group in comparison took the same sort of setting, but took the timeline Mitopure product, and we took blood draws before they took it, 6 hours and 24 hours after. And then this is a crossover design. Basically what that means is that people who took the product, whether it's the juice or the Mitopure, there's a washout period of 10 to 12 days, and then they switch the opposite intervention, and we look at the levels of Urolithin A in the blood with both the interventions. And so what you see is basically the correct answer is most of you guessed with 6 cups. We see this massive difference in the Urolithin A bioavailability in plasma. In green is the level seen with the Mitopure intake versus in red, which is the pomegranate intake. And what you see is about a 6-fold higher, in fact, even 6.8-fold higher levels of exposure to Urolithin A in the Mitopure product compared to the juice. And this is really dependent on not just the diet, but your gut microbiome. And I can personally share that, somebody like me who doesn't have the right gut microbiome, I'm not in those lucky 30, 40%. I really need to rely on products like Mitopure to give me my daily sort of supplementation of Urolithin A.
Thank you, Dr. Singh. So one of the other questions we get a lot is, how do I make the most of timeline Mitopure? How often should I take it? Or what time of day should I take it? So we've pulled together a list of 3 simple tips here. The first thing we'd recommend is create a habit. Take it first thing in the morning with breakfast and really try to take it daily. The second thing is recommended dose is 500 milligrams of Mitopure per day for at least 4 months. Some people may start seeing the effects earlier. We've heard people say that they start feeling the effects as soon as 4 weeks. Others say after 2 months, but if you really want to make the most of it, do take it daily for 4 months. And then finally, in terms of how to take it, how to consume it, we recommend mixing it into a smoothie, yogurt, cereal, oatmeal, chia pudding, or we've also pulled together a list of recipes that taste delicious. This webinar with 4 key takeaways. The first is mitochondrial function declines with age. And that's been linked with various chronic issues, including loss of muscle function and strength. The second is Mitopure is safe, bioavailable, meaning it's absorbed by the body, and it improves mitochondrial signatures in skeletal muscle. The third is 4 months of daily consumption of 500 milligrams of Mitopure are clinically shown to lead to a significant improvement in leg muscle strength. And finally, 500 milligrams of Mitopure delivers greater than 6 times more Urolithin A when compared to 8 ounces or 240 milliliters of 100% pomegranate juice, and that assumes that you can naturally produce Urolithin A. Thank you for your time today and for joining this webinar. If you do have other questions, feel free to get in touch with us via email. You can write to us at care@timelinenutrition.com, and we'll get back to you as soon as we can. Thank you very much.